Biosponge. Can it be used as a biochemical filter media to cultivate bacteria? Biosponge has good permeability, but the total surface area for nitrifying bacteria cultivation is rather small. This time we have ceramic rings. Let them have a contest with Biosponge. If you watched our filter media test before, you may know that effective surface area of a filter media determines how much space is available to nitrifying bacteria. The water permeability of Biosponge is beyond doubt. However, we have measured that the effective pore area is very small. It's the opposite of the ceramic ring. A ceramic ring has a large surface area, but the water permeability is far inferior that of Biosponge. In this test, we will start from the culture speed and the ability of water treatment after the initial culture. For these two aspects, let's see how they perform.
Follow us to uncover more aquarium mysteries.
For this test, 2 identical fish tank drip boxes were prepared. Add 28 liters of water and 2 platies to each tank. Add 1 liter of Biosponge to one tank. Put 1 liter of ceramic ring to the other. Both were totally submerged. Use 2 circulation pumps with the same flow rate. We began with feeding 0.1 grams of fish food per day, monitoring the level of ammonia, nitrite, nitrate. At day 7 since setup, to avoid ammonia level being too high, we reduced the amount of food we fed and continued monitoring. As you can see from the curve, the ammonia in the Biosponge tank has begun to decline, and by day 30, the nitrite had dropped to safe levels. This means that the nitrification system of the Biosponge tank has been established. On the other hand, the ceramic ring tank, the descent speed is much slower than that of the Biosponge tank. So we came to the conclusion, use Biosponge as filter media. The cultivation speed is faster than the ceramic ring. But does that mean Biosponge being superior as bio-filtration media? Under current test conditions, either of the contestants is adequate to stabilize the water of the test. But in theory, for nitrifying bacteria to attach, the larger the surface area, the more bacteria there are, the corresponding ability to treat water will also be stronger. We tried to increase the amount of food we fed, increase the burden on the nitrification system. Let's compare. We set up 2 more tanks. Let's jump to the establishment of nitrification system. Overfeed the fishes. As you can see, ammonia, content of ceramic ring tank, decreased faster than its Biosponge counterpart. This indicates ammonia oxidizing bacteria attached to ceramic rings appears more numerous. We can also see from the nitrite curve, the ceramic ring tank is 1 day faster than the sponge tank to reach the safe threshold. This indicates nitrite oxidizing bacteria attached to ceramic rings also seem more numerous under current testing conditions. Hey, wake up! Time for some summary. Compare the above 2-phase experiments. We suggest that the filter media with good permeability has a fast cultivation speed. In theory, superior permeability can bring more dissolved oxygen and nutrients, facilitates the rapid reproduction of nitrifying bacteria. So when you are setting up a new aquarium, you can put larger portion of Biosponge or other high permeability media to accelerate the establishment of the nitrification system. The larger the surface area of the filter media, the more space for bacteria to reproduce. The more of them you have, the more capable system you get. For high density setup, or in scenario when the space for filter media is rather limited, you may need media with larger effective surface area per volume.